All right, well, welcome everyone to the for March Developmental Disabilities Clinical and Research Rounds. Today we are fortunate to have Dr. Faraj Haddad with us. And I think, Faraj, correct me if I'm wrong, I think you won the award for best presentation a couple of years ago at our research day, which is great. And sort of you've continued on with that, which is great. So Dr. Haddad is an assistant professor in the Department of Physiology and Pharmacology at Schulich, School of Medicine and Dentistry here at Western. He completed his Bachelor of Science in the Department of Pathology and Lab Medicine and his Ph.D. in Neuroscience program, both at Western as well. He worked with Dr. Suzanne Schmidt, who's done presentations for us before, to complete his Ph.D. And focus on using animal models to study developmental and neurodevelopmental disorders. Dr. Haddad completed his Ph.D. in 2021, and he then actually turned away from neuroscience slightly for a bit to complete a one-year postdoc position on a large-scale teaching program, a project focused on uncovering the hidden curriculum. He started at Western again as a faculty member in 2022, so this is his first year, and he is doing research, but also primarily teaching in the interdisciplinary medical sciences program at Schulich. He wants, Faraj would say a minute ago, he really enjoys teaching, but he also is doing research. And so today we'll talk about his work exploring maternal immune activation during pregnancy and the impact on development. And I think this is a sort of increasingly interesting area of research because we know that genetics contributes to neurodevelopmental disorders, but not completely. It's not all about genetics, but there must be other environmental factors, including sort of the prenatal environment. And so I think, Faraj, I can only speak for myself, but hopefully for others, I'm really looking forward to this because I think it's a fascinating area. So Dr. Haddad, welcome, and you have the floor.
Thank you for the introduction. I'm just going to share my screen and make sure that you can see it.
Yep.
Okay, so hello everyone and welcome. So today I'm going to tell you about, again, one of the environmental risk factors for neurodevelopmental disorders. It's maternal immune activation during pregnancy. So just to quickly get started here, I'm just click here. I don't have any conflicts of interest to disclose, and I'm going to just sort of quickly glean over the learning objectives or sort of the things that I'm going to cover in this talk today. So in general, it's going to be a talk where I'm not going to throw a lot of data at you. I'm more so going to tell you sort of the story of how we learned about these, the associations between maternal infection and also the association between maternal immune activation with neurodevelopmental disorders. So when I'm first going to give you a little bit of a background, but then I'm going to talk about the epidemiological evidence that links maternal infection during pregnancy with neurodevelopmental disorders. I'm going to outline some of the preclinical evidence of what we know from animal models that supports the maternal immune activation hypothesis. And then I'm going to sort of just quickly explore some of the sensory processing paradigms that I have used to try and study sort of this topic. And some of these paradigms that have some translational potential for autism spectrum disorders. So let's get us started. What sort of better way to maybe get this started is then with a broad developmental timeline figure. So what you're seeing here in the picture is sort of just something to highlight how much development, how much actually goes on in gestation. So the easiest way to think, to see this is so many of the different limbs are developing even in the first few weeks during gestation. And the really main takeaway that I want you to sort of think about from here is how everything is sort of condensed. So even if something happens for a very short time at this particular point in gestation, a lot of different processes are going to take place. What I also want you to take away is that today I'm going to focus a lot about the nervous system and about the brain specifically and sort of how that plays, how that gets affected. But I want you to keep in mind that whenever we think of something affecting, development, it's actually affecting all of these other organs at the same time, right? So it's in context of a lot of things happening, and it's not just so. Even though we're looking at the brain, it's not the only thing that's changing. And it could be that other changes in the rest of the body can, even contribute to what we see in the brain. So again, the brain is sort of my favorite piece about this. So I'm going to then show you another figure, same kind of thing, but now it's about brain development. And so what this shows is sort of the different processes that happen, throughout gestation and even postnatally after birth and until adulthood, the processes that happen that cause the brain and the nervous system to develop. And so really the key takeaway here is that, we have these certain processes like neurogenesis and neural migration. So this one in terms of, the neurons dividing to get to the number of neurons that we want, or migration, which is for them to go into the place that they need to go. And these things actually are irreversible, right? So, neurogenesis largely, not entirely, stops after this particular period. There are still some, but very limited postnatally. Same thing with neuron migration. Once the neurons go somewhere, they get there and they kind of stay there. Here, I'm just going to enable the pointer really quick. And so again, the idea that if something actually happens during these irreversible events, then the effect of it can be seen or felt for a lifetime, basically. And then, what could actually go wrong, and that affects brain development. So when I like to think of this, I like to think of it very broadly as to sort of what are the different factors that can change neurodevelopment. And, if you really wanted to go as broad as possible, you could think of genetic factors, as Dr. Nicholson just mentioned. And we know that there are, mutations in certain genes. These are just some examples that I've picked out that you may recognize. And when these go wrong, we get sort of, usually it's not just things affecting the brain, but there's a syndrome, and a lot of those, some of the symptoms include things related to the brain. And so these are just some genetic mutations when we have a loss of function in a particular protein. Now, on the other hand, we have prenatal environmental risk factors. Again, it's not necessarily prenatal, so things can also happen postnatally, but at least for today, I'm going to focus about prenatally. And so some of these things are known. We actually, think about them on a regular basis. It's just alcohol during pregnancy, smoking during pregnancy. There's even sort of exposure to drugs. So we know there's a lot of drugs that are contraindicated. You can't really take them, or you shouldn't be taking them during pregnancy. And so we know that these things can affect the brain, and there's a long line of evidence that can support this. But again, the story is never that simple, and there is sort of this interaction between the genetics and the environment. Not every genetic change is likely or is guaranteed. Even in these genes themselves, you can have a mutation that only slightly affects the gene's function and not entirely. And so then it brings us to the idea of the interaction between the genetic risk factors and the environmental risk factors. So today, the majority of my talk is going to focus on this, and I'm going to tell you about infection during pregnancy as one of the environmental risk factors for neurodevelopmental disorders. Excuse me. So the idea of infection during pregnancy influencing neurodevelopment is not something that's novel. This has actually been known. Infection during pregnancy has been known to be associated with detrimental outcomes after postnatally. And this has been known for some time, mainly because of these torch infections. And so these torch infections are, torch stands for, classically, the term came from these particular infections. So Toxoplasma gondii, which is a parasite, rubella, cytomegalovirus, and herpes simplex virus. And really, to summarize this, I actually got this figure from a paper here, a really excellent review article, to show that this figure has two sides. So when talking about torch infections, on the one side is sort of how do these infections, which we know actually can get to the fetus. So they can either, cross over through the placenta. You can also have some sexually transmitted infections that can, be ascending infections over here. But what I'm really concerned with, and this is my sort of area of expertise, lies more along this side of the story, where what actually happens once these infections go up or affect the fetus or fetal development. So again, not on one side, it's not just the fetus that we're talking about, it's the mother. So the mother experiences increased inflammatory response, respiratory distress, in really severe cases, potentially death. The placenta is also affected. So the placenta, there is decreased nutrient exchange. So the fetus gets fewer nutrients. And because of these various changes, you can actually get, growth restriction in the fetus. And you'll see here birth defects, including blindness and deafness, which you'll be familiar with as something that is actually, related to the nervous system. You also get microcephaly or neurodevelopment. So these infections have been associated with these kinds of outcomes. And although the classic infections are these four, the list actually did grow. And again, most of this is from the 20th century to sort of include syphilis as well. But more recently, I wanted to, also highlight this Zika virus over here. So this was a more recent outbreak, if you remember, from around 2016 to 2017, where this was actually associated with a pretty high risk of birth defects. So if mothers who were pregnant and got the Zika virus, their offspring had a high chance relatively of developing microcephaly. And so, again, this is not necessarily the full exhaustive list. There are other infections. In fact, some papers you might read, they'll say the O in the torch actually refers to other infections. But the general idea is that, these actually can affect development in general, but can also affect brain development. So this is sort of how the idea got started. And today I'm going to focus a little bit more on neurodevelopmental disorders. So microcephaly, that can be considered as a neurodevelopmental disorder, but I'm going to focus a little bit more on the things that, aren't necessarily as stark, or we might not see them directly from an early stage. So these are things like attention deficit hyperactivity disorder, developmental language disorders, intellectual disability, autism spectrum disorder, and even schizophrenia. So this one, schizophrenia is a little bit different from these, and that's diagnosed later, but it still counts as a neurodevelopmental disorder. And so, the list goes on, but I'm going to focus specifically out of these on autism spectrum disorder and schizophrenia. Now, this is not because necessarily that these are the only ones that could possibly be associated with maternal infection. The thing is, when it comes to maternal infection during pregnancy, the studies have focused on these two, and we don't quite know how maternal infection, impacts other things, not as well, at least, as these two disorders. And as far as I'm going to be talking about in this presentation. And so because we're going to talk about them, I'm just going to quickly introduce these two disorders and what they look like. So they're actually pretty different from each other. If you ignore the fact that at some point back in history, autism was called childhood schizophrenia, but now the diagnostic criteria are quite different. So autism, typically diagnosed in childhood, presents with social communication and social behavior deficits, increased repetitive behaviors, fixed interests, and more recently also getting acknowledged that it has a lot of sensory processing changes in here as well. So either hyper or hyporeactivity to sensory input. Whereas on the other side, schizophrenia is typically associated with these delusions and hallucinations. It tends to present later in adolescence and maybe early adulthood, and it has, positive symptoms and negative symptoms. There's also what you can't see, cognitive symptoms that are pretty well known in schizophrenia. So these are quite different. So what actually brings them together? When we think about these two disorders, they are actually both quite prevalent. So if you consider that schizophrenia generally affects 1% of the population, autism, the diagnosis has gone up in recent years, up to 2% potentially. And what's really also brings them in common is that they're pretty difficult. We don't understand them very well. We know that genes are involved. We know that the environment is also involved, and gene-environment interactions are both at play as well. Now, also, because I'm going to be talking about maternal infection, I do want to highlight that both these disorders have been associated with inflammation. So individuals with these disorders actually have more inflammation in general in their bodies usually. And what I'm going to tell you today is that they've both been associated with maternal infection during pregnancy. So in this first part of my talk, I'm going to take you through the story of the epidemiological evidence that linked maternal infection during pregnancy with autism and autism spectrum disorder and schizophrenia. And so to do this, I decided to sort of play everything out in a table. There's going to be a lot of information here, but I'll help orient you a little bit. So in terms of exposure here, we're going to talk about what is the kind of exposure, as in what kind of infections are we talking about. Everything on this table has been associated to some degree with schizophrenia in the offspring. Then we're going to talk about whether there's specific pathogens and markers, which populations, so where were these studies performed, and the references in case you wanted to sort of take a look. And all of this was reviewed in this review article that I published a few years ago, so you can actually go take a look, and there's a whole table in the supplementary section that can show you all of these evidence in one place. So the first round of studies that actually found this association is that they kind of looked at individuals whose mothers were pregnant during periods of epidemics or pandemics. And in here, we don't actually know. So, I mean, it's assumed that these individuals may, if they were pregnant during that period, that they may have contracted the disorder, the infection. So a lot of these you'll see this, in 1957 is a common one. Even the second study actually spans 1957, and that's because there was an influenza epidemic. In some cases, it's referred to as a pandemic in 1957. And so what these studies noticed is that individuals whose mothers were pregnant during the pandemic, not knowing whether those, they were infected or not, those individuals had a higher risk of developing schizophrenia. Now, then we're going to go, basically every row is going to get a little bit more specific. So later on, studies actually started looking at the clinical records. So are there records of people coming into the hospital or, going to their doctor and when they were pregnant and having an infection. So in this case, we get a little bit more specific because now we know they could have had a respiratory infection or a genital reproductive infection or influenza. And so here we basically, or you could, maybe say a viral infection. So here the studies were conducted a little bit later, you'll see, and they noticed that if there is a clinical record of infection during pregnancy, then the offspring had a higher chance of developing schizophrenia. Then more specifically, we started getting serological evidence. So now we actually can detect these particular, so they could detect the influenza. So they know that during pregnancy, there was influenza virus in the body, in the mother, or Toxoplasma gondii, which is one of the parasitic infections. And once again, they showed an association with offspring, the risk of them developing schizophrenia. And finally, the most specific studies we have are those looking at what happens after infection. So whenever you get an infection, you'll probably, how you feel crappy, you get a fever. That's usually caused by these inflammatory markers, cytokines and chemokines. And these are just some examples of them. And the really key thing about these studies is that they actually saw that the level of the cytokine, or the immune marker, so this is, for example, C-reactive protein, it tends to go up when there is inflammation in the body, is that the higher the level during pregnancy, the higher the risk that an individual had to develop this disorder. And so this kind of summarizes the studies, or summarizes studies. Obviously, this is limited. There's other studies that have also looked at this that I'm not showing over here. But then I'm going to go into very similar for autism. Now, first off, before I go through there, you'll notice that all of these studies sort of progress and build on each other. So from the epidemic or pandemic studies to the clinical records to the infections, the timeline sort of is getting closer to now. And that's what you'll also see with autism. So in fact, as far as I'm aware, there isn't sort of a a study that has looked at the risk for autism based on whether the mother was pregnant during an epidemic or pandemic. But we do have those that looked at clinical records. So again, we know roughly viral bacterial infections, even fever during pregnancy was associated with an increased risk of the child developing autism spectrum disorder. And then same thing with serological evidence. So then we can see here some of the torch infections. Remember the same one, cytomegalovirus, herpes simplex virus. And finally, with the pro-inflammatory markers, again, we see that, C-reactive protein, interferon gamma, different interleukins, that these are increased in the mothers. And when they are detected or increased in the mothers during pregnancy, then there is a higher chance of the offspring developing autism. And so then, what I just gave you is sort of a very quick breakdown of which studies have shown an association, but that's not the full story. There are other important things that we have to take away from these studies. So some key takeaways from these findings is really the importance of timing. So we just kind of talked about how, there's a lot going on during development. And what these studies show is that the earlier that the infection happens, typically in the first to second trimester, early to mid gestation, then that's where the risk tends to be the highest. And this is, you can think of it as, in the first little bit, you're laying the foundations of development. So the earlier you introduce something that disturbs development, the more things are actually messing up. Your foundations are not very solid, and everything you build on that in terms of the brain is going to be a little bit more shaky. The other really key thing to think about is sort of the strength of the association. Now, I just showed you there's so many studies that have actually looked at this. And you're wondering, we're screwed. Everyone's going to have this increased risk of neurodevelopmental disorders. But in fact, what we see is that the ratio, so the odds ratio, that the increase in it after an exposure to infection during pregnancy tends to be small. So maybe between factors of 1.5 to 2. So this isn't a cause of neurodevelopmental disorders. It's more referred to as sort of a priming factor, something that increases the risk further. And if you have multiple risk factors, each of them are just a little bit, they can eventually push someone over the edge and cause and lead to the disorder. There's also the idea of severity. So I referred to this a little bit when we're talking about the inflammatory markers, but the higher the severity or the higher the fever, the more intense the infection, the more the risk that is associated with it. And then we have this idea of infection type. I showed you a table, and for each, if you remember, for schizophrenia and for autism, there's so many different ones. So I'm just going to quickly say I'm referring to viral infections with this little icon here, bacterial infections, and then finally parasitic infections. So the idea is that all three of these kinds were associated with both the disorders we're talking about. And so this kind of says that it's not necessarily something that's very specific to a particular infection, unlike the Zika virus. And we'll touch on this in a little bit as well. Finally, very few studies, not many have actually looked at this, but there's a couple of studies that have shown that a family history of these disorders makes someone more susceptible to the risk. So the risk is higher in those with some kind of family history of neurodevelopmental disorders. And then the last thing, and something that I didn't actually cover in the tables, is that now the studies have moved on, or not moved on completely, but some of them have actually started investigating how does infection during pregnancy actually change behavior. And so this is thinking about autism spectrum disorder, for an example. We know that sort of there's a spectrum of different symptoms, and different individuals experience different things. And so the idea here is not necessarily whether infection during pregnancy increases the risk of a diagnosis, but does it actually change how someone behaves in a particular way? Does it change their social behavior, even if they don't have a diagnosis of autism or schizophrenia? So that's where some of the more recent studies have focused. Now, again, with any kind of field, there's also some important considerations to keep in mind. The first is, many of you who are familiar with these kinds of disorders, is that diagnostic criteria change over time. Okay, and so we're not necessarily, the studies that were done in 2000, they were using a diagnostic criteria that is different from the studies that were performed in 2010. And even the individuals that are diagnosing these, maybe they were diagnosing these disorders back in the 80s or 90s. And so a lot of these studies are retrospective. So we do have to keep that in mind. But as you saw as well, the populations we're looking at have spanned quite some time, and the associations are still there. So there is some important effect that we really do need to take a look at. The other important consideration that I want to sort of point you to here is that the findings are actually limited to certain countries. So if you'll notice, most of the countries I actually showed were either in Europe or in North America, or basically the States. So we don't actually know how this looks elsewhere in the world. And I would say that's a pretty substantial limitation in what we know about this topic. Also, sort of depending on the study type, there are some studies that have collected a lot of data, and that kind of gives us more of an information, whereas some studies, like, there aren't as much data for us to actually make these associations. And taken together, it's actually difficult to isolate the effects of maternal infection, whether what we're seeing is sort of a risk because of maternal infection during pregnancy, or whether there's other factors, both prenatally and postnatally, that we haven't accounted for. And so this way basically brings us to the importance, or, it led researchers to try and look at this in a rodent model where they can isolate the effect of maternal infection and see whether that impacts the offspring and their neurodevelopment and their behavior. And this is what I'm going to talk to you now. In my work, I did a lot of behavioral studies in animals, in preclinical models, trying to understand how this works. And you'll see that now I stopped talking about maternal infection and I now start talking about maternal immune activation. So why is that? So whenever we talk about maternal infection during pregnancy, whenever we talk about the effects of any infection, the outcomes that we get, I kind of like to classify them broadly into two camps. The first camp are these pathogen-specific effects. So these are outcomes that are specific to a particular pathogen or sometimes a group of pathogens. So, for example, a virus may come, a certain virus will bring about outcome X, a certain parasite will bring about outcome Y, and so on and so forth. So this is what you can think of about the example of the Zika virus. Microcephaly is an outcome that is specific to some pathogens, very especially Zika virus. It's not something that you see as an outcome to, across all the different infections. On the other hand, there are these pathogen non-specific effects. So these are things like what I'm talking about in terms of autism spectrum disorder or schizophrenia, where they're associated with different types of infection during pregnancy. Now, this basically suggests that this association, and again, I'm not saying that these two are mutually exclusive. You can have both pathogen-specific effects and non-specific effects in the same case, as we saw for example, cytomegalovirus and so on. So, however, this side of the story kind of tells us that there's a common factor. So there's a common factor between the different infections. And if you think about, you've probably had a lot of different infections in your lifetime, even if whether it's a bacterial or viral infection, an outcome that's related to most of them is fever, right? You can get a lot of fever. And the reason why fever is associated with multiple types is because our immune system is evolved in a way that it uses sort of similar mechanisms to fight off different infections. And so fever is caused by some of these, what I mentioned is these inflammatory markers, the immune molecules. And because of that, this led researchers to hypothesize that it's actually the maternal immune response, hence called maternal immune activation. That's what's causing this particular group of outcomes or disorders. And so this is where the preclinical world, the animal studies have really focused on. They really want to answer this question. Are pathogen non-specific effects or maternal immune activation linked to neurodevelopmental disorders, or do they lead to changes in brain development specifically? And so this kind of, there's been a lot of studies done on this. What does a typical preclinical model for MIA look like? And I did this in my research. Typically, what it looks like is some animals that are paired together. So after conception, the female gets pregnant. The female is then exposed to maternal immune activation sometime during gestation. Now, the time specifically, obviously, in this case, the example is rats. The time is picked so that it coincides with the human equivalent of the developmental stage at first to second trimester, because that's when we see the highest risk in human studies. Then the animals give birth and the offspring are then tested. So we're not testing the mother, not always, but we're testing sort of the offspring once they grow up, or when they're young, or when they're really old. It all, it depends on the kind of study. So I just want to focus a little bit here, because talking about maternal immune activation, because you've probably never had an infection. You've probably never had an immune response in the absence of an infection. Or I would say, I guess there are many other kind of causes, so I won't overgeneralize, but typically we think of immune responses as being related to an infection, right? So how do we induce a pathogen-free immune response in these animals? So first we have these receptors that exist on immune cells called toll-like receptors. These receptors have evolved throughout evolution to detect things that look like infections. So things that look like a virus or things that look like a bacteria. So here's sort of an example. You'll see these different receptors here, different kinds. And with each one, there is a particular molecule that binds to it. So these molecules, the ones at the top here that I've listed, these tend to be associated with bacteria. So one common one in MIA models is this TLR4 agonist lipopolysaccharide. So this is actually part of the outer membrane or cell wall of bacteria. So as soon as the body sees it, it knows I need to induce an immune response. Same thing with viruses, but here now you can see them more on the inside of the cell because they get brought inside. So double-stranded RNA, that's not something that we usually have in our bodies. That's usually associated with viruses. So we can inject either LPS or this other one, poly-IC, into animals, and that leads to inflammation over some time. There's no infection, so the inflammation goes up for a short amount of time, and then it goes down. So this helps us really control how much inflammation is happening, and without sort of the pathogen in there, risking sort of a different response across different animals. So what happens afterwards? The animals are born and then they're tested. So as you can see here, the tests that have been done span a broad range of techniques. So on the one hand, we have molecular tests, so we can look at the level of neurotransmitters in the brain. So glutamate and GABA, glutamate activates, it tends to activate cells in the brain, and GABA inhibits them. Studies have also looked at the cellular changes. So what you're seeing here are immune cells in the brain called microglia. And based on their shape, we know that their shape is associated with a change of function. So if they look a little bit more plumped, that means they're actively cleaning up things around them, and actively eating up different pieces of the neurons, and in terms, not eating the neurons themselves, but sort of eating up some of the connections to make the connections more refined. We've also seen electrophysiological changes. So, studies have tried to look at the firing of neurons, so how active these neurons are in different brain regions. So what you're seeing here is sort of each color is a different brain region, and you can see how the brain regions are synchronized in their activity. So this synchronization is important for proper brain function. There's also studies that have looked at anatomical changes. So whether there's whether there's an increase or decrease in brain volume, or the gray and white matter in the brain. Behavioral studies are actually the majority of studies include behavior. The reason is because how do we diagnose these disorders? It's based on behavior. So what you're seeing here is some rats, so showing some social interaction. And then finally, because it's an immune change, we actually look at the immune response as well. So there's been over 400, at least as far as I know, 400 primary research articles on this topic, and the list is growing rapidly. So I'm just going to quickly summarize some of the changes that we've seen. This is, again, I'd love to talk about the data more, but for the purposes of this talk, we're not going to go into too much detail. But, on the molecular side, we know that there's a change in excitation inhibition balance in the hippocampus and prefrontal cortex. We know that actually the offspring that are in these studies that are exposed to maternal immune activation during pregnancy have changes in their immune system, in their gut microbiota. There's also changes in dopaminergic hyperactivity, so they are more responsive to dopaminergic signals. On this middle side here, with these kinds of changes, we see there's an increase in inflammation in the brain of these offspring. There's changes in brain volume, changes in layering of cortical neurons. So these neurons actually travel, as we said, they migrate from where they start when they're produced to where they actually end up for their function. And this change in layering actually tells us that their migration is affected. There's also a change in the synchrony between different brain regions here, in this case, the hippocampus and prefrontal cortex. And finally, for the cognition, we have changes in, working memory, cognitive flexibility. More specifically to autism, there's changes in social behavior and communication. And even, again, more specifically here, changes in sensory processing and stereotypical behavior. So there's a lot of actually studies that have looked at this, and that we know that MIA during pregnancy does affect the brain in a lot of different ways. But how does it actually do this? And before I go into it, I forgot I also had this. So the research on this topic has actually progressed so much that there are now non-human primate models studying this change. So the rhesus macaques are sort of the top dog of all neuroscience research. This is where the closest usually that you can get to humans in terms of cognition and behavior. And this one particular group, they saw changes in behavioral flexibility, and they even saw changes in brain volume. And then, this is again, you can appreciate that there's a lot of work and time, because the time it takes for these animals to breed and deliver is much longer than in rats. So what is actually causing these changes? So I, again, took this, this is one of my favorite review papers about the topic, and I want to highlight focus your attention on a couple of things. So first off, because we said these offspring have a change in their immune system, the peripheral immune system, if it's changed, it actually plays a role in sort of determining what kind of microbiota live in the gut. And over the past few years, we have understood that the gut microbiome, these bacteria that live in the gut, actually influence behavior and influence the brain. So that's one way that, we can influence the brain. On the other side, this is where I'll direct your attention, is that you'll see these cytokines. I mentioned cytokines, these pro-inflammatory markers. So because the immune system is changed, or even during pregnancy, when the mother gets this immune response and then releases all these cytokines can get to the fetal brain, and they can either get to the microglia, which are the immune cells of the brain, the neurons, which are here, or the astrocytes. And they can actually impact a lot of different processes that I showed you way in the beginning: the neurogenesis, neuronal migration, the pruning of different connections between neurons. And so that's sort of the main mechanism that is thought to influence or cause these different changes that we see in the brain. Now, another recent topic as well I wanted to highlight is this is a really prominent figure in the field of maternal immune activation. So if you really want to see sort of the historical overview of the field, you can just go to this person's publications and sort by date. Is this idea of resiliency versus susceptibility. So it's not just that maternal immune activation happens in isolation, it happens in context of other things. And you can see here there are factors that, if the mother has gestational diabetes, if the mother has anemia during pregnancy or experiences maternal stress, all of these things can actually make the child more susceptible to maternal immune activation if it happens. And on the other side, you can also see how these other factors then promote resilience. So, all of this evidence, a lot of this, what I'm telling you, is from animal models. But if, for example, the mother has a particular diet during pregnancy that, adjusts the immune system a little bit, or the immune responsiveness, then you give, when the mother experiences maternal immune activation, the effects on the offspring aren't as severe. And so this is where I'm going to sort of take away. So what you see on this figure is all environmental risk factors, so things that happen in the environment. So what I actually wanted to study as well is this genetic risk factor component. And this is what I actually looked at in my last study that just got accepted for publication. So here I'm going to tell you very briefly about some of the work I did that investigated the interaction between an autism spectrum disorder risk factor and maternal immune activation. And I'm going to focus a little bit on, sort of the pre-attentive sensory processing. So bringing it back to this sort of idea of genetic risk factors and environmental risk factors, I'm going to be focusing very specifically on this interaction, because as Dr. Nicholson mentioned, if we take all the cases that, all the cases of autism that are associated with a genetic, with 100% with a genetic mutation in one gene, those account for only a small percentage. So there's really a missing piece of the puzzle that we aren't understanding. And the only way we can figure this out is to better understand the environmental risk factors and the interaction. And so in our study in the lab, in Dr. Suzanne Schmidt's lab, we actually worked with animals that have a mutation in this CATNAP2, contactin-associated protein-like 2 gene. So this gene and the association between this gene and autism was first discovered in 2006 in this paper, where they noticed that this one population of old order Amish individuals had a recessive mutation that loses the function of this, that leads to loss of function in this gene. And they noticed that, again, it's not specifically, it's not just autism, but these individuals have intellectual disability, epilepsy, and language impairment. So actually this language impairment is something that is pretty stark in some cases. Individuals develop language, but then they regress and lose that ability. So what does this gene do? It codes for a protein called Caspr2, and this protein is involved in these various functions. If you remember our friend neuronal migration that we mentioned, that's something that could be irreversible if it, if it's because it happens early on in brain development. So what we decided to focus on in the lab, and this is sort of what the focus of Dr. Schmid was, is sort of this particular piece of the puzzle, the hyperreactivity to sensory input. And so why did we decide to focus on this? And to actually take you through, I'm going to take you through an analogy. So you may have come across one of these Where's Waldo pictures. And this is, for me, what I call sensory overload, which is, there's so much going on that it's hard to actually focus and try to find Waldo. And if you're wondering, he's right here in the middle. And sort of there's this idea of sensory overload, but when our brain actually encounters a lot of sensory information, as in this case is all visual, but it can also be, auditory, it can be of various different modalities. Our brain actually has processes that help us filter out the redundant stimuli. So it kind of looks like a little bit like that filter. You have a lot of different stimuli, and then the brain will choose the ones that are most salient, and then those are the ones that it's going to process. Now, it's thought that, in individuals with disorders like autism and schizophrenia, that this sensory overload can underlie impairments that are seen. So if you think about a social environment and your brain just can't quite tone out the sound of a fan that's in the background, that might actually distract you, and it could make you not be able to hold up a conversation just as well. And I just kind of wanted to mention that it's the brain, it is doing the filtering, but it's also the brain that's then processing the information that comes out the other end. So how do we actually study this in animal models or even in humans? So Dr. Schmid worked a lot with, and just before I kind of, to illustrate this a little bit better, so in a typical individual that looks at this person, which is a lot less busy, this doesn't have as much sensory input. But to someone who has impairments in sensory processing, when they look at this, it might feel like they're looking at this picture because their brain can't process, can't filter out some of the unnecessary information. And so the way we studied this is through the acoustic startle response. Now, the really main selling point of the startle response is that we can actually measure it across species. So what is the startle response? It's basically if someone slammed the door behind you, you would have startled, you blink, you kind of get this little twitch response, and it's a defensive, evolutionarily conserved response. We can also measure it in rats pretty easily. So the rat is sitting on this sensitive platform, and as soon as they startle, we can measure that displacement. So what does this actually tell us? How does this allow us to measure sensory processing? Well, we can actually look at how much that the animals respond. So or individuals or humans, whenever, based on different kinds of stimuli, so whether it's sounds are loud or quiet, do the individuals startle a lot? So if they startle a lot, then that sort of tells us that they react more compared to the other individuals in terms of to the same sound. On the other hand, you can also think of when you see someone reacting more, it could be that they just, they're inherently more reactive, but it could also involve this idea of sort of sensory filtering, where habituation and sensitization. So you've probably not, it's very rare that in our lives we experience a sound only once, right? You've heard the sound of your microwave many different times. So what happens after you hear that sound over and over again? So typically, after exposing individuals to the same startle stimulus repeatedly, they habituate. So the response actually goes down. However, in some cases, think of your fire alarm, for example. If your fire alarm keeps going on, you're not necessarily going to habituate, you're going to sensitize. It's going to bother you more and more. So there's this idea of, when we see someone with an increased reactivity to sensory input, did they actually start off that way, or did they just sensitize or habituate? Did they sensitize to this response over time? So we have there have been quite a few studies, particularly by this group of researchers, that have shown pretty robustly that in individuals with autism spectrum disorder, the sensory reactivity is increased. What about sensitization or habituation? It's not as conclusive, the evidence, and there's more considerations about how things are measured, but we pretty much know this is the reactivity increase. So then the other question is this increase in reactivity due to sensitization? And that's kind of one of the interesting questions to know. So what did we do to test this? We got these animals that had, one functional copy of the catnap2 gene. We bred them together, induced immune activation during pregnancy, tested them when they got older. And then, because these animals had one copy, then their offspring could either be normal, wild type, with two functional copies, heterozygous with one functional copy, or homozygous knockout. So this is a complete deletion of this particular gene. And so usually we see that, and then within each group, we also have those that were exposed to poly-IC during pregnancy and those that weren't. And so usually what we wanted to see was, these individuals have a susceptibility. These animals have a susceptibility. They only have one functional copy. So if we expose them to something more, will they actually become impaired? Typically, these ones don't show any change. And then finally, for the homozygous knockouts, we have repeatedly shown that these animals actually showed some severe phenotypes. So can this get worse if we expose them to maternal immune activation? So I'm just going to give you just one example from the study. Again, you'll be able to see it pretty soon. But we tested animals on 30 trials of the startle sound at 100 decibels. we sort of looked at how they sensitized. So usually animals will sensitize right after the first trial. They'll sensitize quite a lot. And we wanted to see whether this degree of sensitization was increased based on the genetic component and the maternal immune activation component. So what you're seeing now in this graph is sort of the adolescent male animals, the sensitization score, which is the ratio that we've calculated here on the y-axis, and then with the different genotypes on the x-axis. And what you see here is in adolescent males, the sensitization increases due to the knockout of the gene, but not due to the maternal immune activation. Now, when we look at in females, this actually isn't the case. We now see that in animals are sensitizing because of the poly-IC exposure during pregnancy, but the genetic knockout doesn't actually affect sensitization. And that's just part of the story, because then when we follow up animals and look at them in adulthood, then we don't see any effects due to genotype. So basically effects in between here. We only see an effect due to the maternal immune activation, and it's still a broad effect across all groups that sensitization gets increased. So this is just one of many different paradigms we performed. I'd be happy to chat a little bit more about it, but what can we take away from these studies, right? Obviously, these are animal studies. There's some translational potential because startle can be measured very similarly to humans. But what this tells us is actually that multiple risk factors, so maternal immune activation or a mutation in CATNAP2, can actually be contributing to the same phenotype. So in this case, it was increased sensitization. So when an individual presents with an increased sensitization phenotype, even if they have a mutation in the disorder in a particular gene, that doesn't mean that other factors aren't also contributing to that phenotype. The other thing that we can take away from this is that the way that a risk factor influences sensory processing or other phenotypes actually depends on what we're measuring. So we're measuring sensitization. If we look at another phenotype like just reactivity before sensitization, that shows a completely different pattern. And that actually changes depending on sex and age as well. So these are just some implications which can actually have some, importance clinically. Also, whether you have reactivity increased because you're sensitizing to something, or whether you just have that at baseline, that might be also important to know. So that's kind of the end of the road for this particular topic, but where do we go from here? What is the future of this field of maternal immune activation? Now, you probably thought the first thing when I said infection during pregnancy, and you may have thought, what happened because of COVID? Because COVID has affected millions, if not billions of people. Are we going to see a surge in these disorders in 5 or 15 years from now because of this? And the answer isn't as easy as initially thought, right? Because there's many factors, as we saw, that can contribute to resiliency and susceptibility. If you also think about, a lot of people were at home. So because they were at home, they weren't exposed to all these other infections. And even if they did get COVID, then they may have been less likely to get another kind of infection. So when we look at the overall diagnosis or prevalence of a disorder, it might not go up that highly. The other thing is that this might actually be a blessing in disguise, because now we have a lot more data to study this topic in more detail. And then the previous epidemiological data was quite restricted in space and time, but now all of these other countries that we've never looked at before, they probably also tracked COVID. And really the main takeaway that I want to say here is that really more prospective studies are needed, because a lot of the time we're very reactive. But the best way we can actually figure out these associations is to plan studies proactively and actually try and measure and follow up people over some period of time. This is still an active field of research. There are many outcomes and mechanisms being investigated. This may help us uncover, how these disorders come about, or it may actually also help us uncover some interventions that could reduce the impact of maternal immune activation during pregnancy. And with that, I'd like to thank, mostly a lot of this work was done in the Schmid lab with Dr. Suzanne Schmid and Dr. Cleosa de Oliveira, who, were sort of really leading this and are the other co-authors on the recent paper. This is an old picture from the Zoom times right after the pandemic. Thanking some of the collaborators. Here's my contact information, all the funding sources that this could not have been possible without. I'm happy to chat more about the data, and even again, if there are any individuals who do this kind of work or have access to this kind of data in human, in clinical settings, I'll be very interested to sort of chat about opportunities for collaboration. And that is all. Thank you for listening.
Thank you so much, Faraj, for that. That was a fantastic review. I mean, it really summarized the field nicely. Just a quick thing. So I think a woman by the name of Stella Chess back in the 70s or 80s did research on autism and the increase following, I think it was a large scale epidemic of rubella in the 60s, and showed an increased rate of kids with autism following it. But I think they probably would have been very, very disabled kids because of what rubella does to the fetus. And so I'm not sure it would be the same as what we think of as autism now. But regardless, I mean, that was one thing. The perhaps though, if I could take the first question, then if people want to ask questions, you can use the chat if you feel, or the Q&A. Preferably, it'd be nice for people to use the raised hand functions, because then we can actually have discussion of things. But Faraj, where is your research going now?
So currently, because of my my position is focused more on the on teaching, so I don't have a wet lab. So I'm probably going to be moving away from the animal work or preclinical studies, besides some potential collaborations. But I am interested in sort of pursuing this and going more into the study, trying to collect some of this data and seeing how these risk factors during pregnancy impact or, change the risk in the offspring. So something more on, the human studies side. It is more, I think that's where a lot of, there's a lot of preclinical evidence, but to translate things, it's much more difficult to move. Again, as you can imagine, the immune response, we're studying just the immune response on its own. How does this actually look in humans with the infection? What are the different components of the immune response? Which of these cytokines are elevated? These are really important. And the best way to try and do this is to sort of go with prospective studies where, really a lot of this evidence came from, if you notice in the tables, there was a cohort from the States between 1960 and 1967, where they recruited a lot of people and they followed them up. They collected samples from them. So I would actually love to be able to sort of do this one day. So I'm trying to see if there's someone who's interested to take this on with me. It's a very long-term thing, but we'll see where it goes. So the group that I work with, the Province of Ontario Neurodevelopmental Disorders Network. So we actually have been working with somebody who was at MAC. I think she's at University of Texas now. Jane, can't think of her last name right now off the top of my head. But and so I think what the immunologists who are interested in this, we're looking at was, I think there's a woman at Stanford or somewhere in California, Judy Vanderwater, I think her name is. And she's shown an increased rate of certain cytokines and autoantibodies in the mothers of people later diagnosed with autism. Now, the blood work was taken many, many years later, right? So it wasn't taken at the time of birth, which is a real problem. But do you know much about that work? About autoantibodies, I don't know too much, but I can tell you for sure from going to one of the conferences is that the CatNap2 gene that I'm talking about, there is active research on that particular one, where if there's antibodies against the protein, the Caspr2 in the mother, then that is actually associated with autism. So that's a whole other sort of, it's funny because the two fields don't talk to each other as much. I didn't find out about that until going to the conference, but the autoantibodies against that protein are actually associated with autism there.
But that's a shame, really, the two fields. I think it's the problem everywhere, right? With people working in their own area, but not sort of cross collaborating or because obviously it's not all genetics and it can't all be immune, presumably, and so there's got to be some predisposition that people have, which is presumably this is then sort of affected by other environmental factors. So I guess I'll go out on a limb and say it's both environmental and genetic, but it's surprising, unfortunately, you don't see people doing those kinds of work like you just like you just mentioned. All right, so there's a question here from Genevieve Easterbrook, and she asked, there are lots of fascinating avenues to explore. What about non-infectious inflammation, such as what is seen in obesity during pregnancy? Are you familiar with any of that work?
So what I would say is that in terms of obesity, and again, I'm more familiar with the preclinical work, so I can answer some of your questions. So if you remember that figure that showed the susceptibility and the resilience, some of these studies are actually from mothers, and one of them was gestational diabetes, and I think some of these actually have to do, when they study them in mice and rats, they change the diet, and that does actually cause like a weight gain and things like that in the animals. And the other aspect also on the other side, there's something that that promotes resilience that also has to do with diet. So that's the link from the preclinical work. Yes, there is increased inflammation. Now, the one thing that I would say is, to really be able to answer this, we would need to see sort of what are the cytokines involved, because we know, so from the maternal immune activation work, we know that, for example, interleukin-6, if you that is really key, and that actually can affect the brain development. So one way to look at this is to kind of see what are the level of these inflammatory cytokines in obesity, and that could provide some kind of link between them. That's kind of as much as I can answer from the preclinical work. Again, because the environmental studies or the epidemiological studies haven't followed up people very closely. So I know a lot of them, they adjust for family history sometimes, or age at pregnancy and things like that. But I don't recall any that have adjusted their epidemiological findings based on that particular status. And then for your other sort of point about the placenta, so this was actually one of the cases in another study that we did. We wanted to look at the immune cells in the placenta. So there's a basic scientist in the Department of Anatomy and Cell Biology. He studies the placenta closely, and he uses the same model that we used, which is the poly-IC model, to study other kind of outcomes such as growth restriction in the fetuses, and also relating it to preeclampsia. And so in that case, that I can't recall at the top of my head in terms of what they have found in there, but that is very true. And one really key component is that the placenta has immune cells, and those immune cells can themselves amplify the signal that is felt by the fetus. And so basically what happened is there is one key study that showed it is the placental immune cells. So if you give this maternal immune activation and see the effects, but then you block off the action of the placental immune cells, the effect of the maternal immune activation goes away or gets reduced. So in some case, the immune response is not just what happens in the rest of the body. The immune response in the placenta itself can actually play a key role in these effects.
And so, Faraj, along with that, Genevieve, I don't know if you saw her comment there, but she collaborates with Steve. I don't know who Steve is, but that's fantastic. But was suggesting you should all talk, which is fantastic. That's what we hope to encourage by these kinds of presentations. Yes, Faraj, question for you though is how much specificity is there in terms of the effects on the sort of fetus if mothers have infections or have this sort of immune activation? So in terms of the offspring outcome, right? Or is it more than just generally, like, is there any specificity? So is it just autism and schizophrenia and intellectual disabilities, or what do we know about other conditions?
So there's some limited association with, I think some studies have tried to look at the association with epilepsy and with bipolar disorder. So, and again, it's not as well understood. There aren't very many studies that have looked at those, and whether the infection whether it's a infection-specific outcome, we are only able to make this conclusion that it is not just the infection, but also the maternal immune response, because we have a lot of studies that have looked at a lot of different infections. So if you think about another disorder, for let's say, epilepsy, and someone wanted to look at that, the first little while they're going to look at they might look at a specific infection and say, oh, it's this infection that increases the risk. But as more studies happen, and we see the association with other types of infections, only then can we, separate out what is specific to an infection or versus what is more general, and it's probably more likely the immune response that's causing it.
Because I think there's a guy at Johns Hopkins, Bob Yokin, I think, who's done work on schizophrenia, and I'm not entirely familiar with it, but I think looking at the idea of the prenatal infection as well. And so I think it's probably been examined in other conditions with the idea, again, that it's not purely genetic factors. Which sort of you had summarized with what you had talked about earlier on. Are there other comments or questions or statements or whatever for Faraj in this area that I think is really a burgeoning area of research in neurodevelopmental disorders?
And I'm always welcome to answer questions outside of this. So feel free to email me if you have any. Yeah,
and I think Faraj's email address was on your last slide there, I think, as well. But, Faraj, thank you. This was a fantastic summary and really nice review of sort of where the field's at and what the findings have been and where it's going. And I really appreciate you coming and joining us today. I think it was wonderful. And we look forward to hearing about where you're going with it in the future and what happens.
Thanks for having me.
No, our pleasure, and thank you. So just a reminder for everybody that please, you'll receive an evaluation. Please fill it out. If you have topics for future rounds that you would like to see happen, please let us know, and we can sort of do our best. The annual Spring Continuing Professional Development Day in Developmental Disabilities is on March 22nd, 2023. So a few weeks from now. It is in person at the Ivy Institute, I believe, and up on Windermere. And there's lunch for everybody who registers in person. The event at 1. It can also be, you can also register online to attend online, but there's a free lunch at noon if people want to attend in person. And our speakers are going to be Mary Jenkins, who will be talking about movement disorders and people with developmental disabilities. Caitlin Castro, I think, is going to talk about transitions from child and adolescent services to adults. And then Jenny Wells will talk about aging in people with developmental disabilities, particularly, I think, Down syndrome, which is a really fascinating area. And then a month from now, for our monthly rounds, Sean Veerakumar, who is a PGY 6, I believe, or PGY 5 in psychiatry, is going to talk about self-injurious behavior. And we look forward to that. But Faraj, thank you. This was a wonderful talk. Appreciate it greatly.
Take care.
All right. Take care, everyone. Bye. Thanks, Faraj. Thanks, Jude.